Today we are going to talk about the topic of horizontal gene transfer, or someone called lateral gene transfer. It should be equivalent term in a broad sense. And we hope to reserve again some time on the second hour of the lecture and also the third hour at noon to have a laboratory practice. And today we are going to use Mr. Bayes and to reconstruct the phylogeny. And if we have time or anyone are interested, we can practice a little bit about PAMO if you like. We'll see. So let's move on to today's topic. So basically, the definition of horizontal gene transfer, or in short HGT, can be defined as a transfer of genetic material from one genetic compartment to another. So that means that it could be within a cell or between the cells, and it's just depend on the different definitions. But usually we only concerned about some particular types, but not others like hybridizations, and which is much more frequent in many organisms, including plants. And we are not going to focus on that. And we basically can categorize the HGT into three different categories: interspecies HGT, intraspecies HGT, or within cell HGT. And there are many cases for all the different kind of categories. And for the interspecies HGT, of course, including those hybridization events. And but in this case, we will talk a little bit more on the lateral gene transfer in bacterial genome. And at the end of this today's lecture, and since right now we have a lot of information on that, and although this is a very common phenomenon for bacteria, but right now we can use a little bit more larger scale to examine the HGT. And for bacteria, to take up the genes from other places, there are three different mechanisms for that, and we are going to talk about that in a minute. So for such kind of genetic material to be transferred, the basic mechanisms can be listed here. And one is, of course, is due to like hybridization events or the transformation conjugation in bacteria. And after the hybridization, there is some recombination of genomes, and sometimes the only part of the genome will be taken up into your own genome, and sometimes it's a huge combination. So it's also depend on the different scale, but this is a very common mechanism. And the other also happen in hybridization is the mitochondria or chloroplast capture. And this is a special phenomenon that the whole mitochondria or the whole chloroplast was taken up from outside, from somewhere else. And in this case, because in a particular cell, there are always quite a lot of organelles, like tens or hundreds of organelles in a cell already. When you take up some of the organelle from outside, if you want to repress it, it's possible that due to the lineage sorting, we could take up some or the other kind of organelle. And in some case, which is well known, like the mitochondria capture, and it was due to the transmissible cancer, and which is a special cell line. It's a cancer cell line that can be transmitted from different cats and dogs and so on. This is a famous example for CTVT. And because the cancer cell line, the mitochondria is kind of deficient in a way, so it's very likely to degenerate. So it needs to take up the host mitochondria frequently to incorporate the normal function. That's one thing. And in some cases, there are hybridization event, and also and after that, there's chloroplast captured in plants, and the whole chloroplast will be taken up by also due to the lineage sorting to take up one of the parents from the hybridization. Or in this phenomenon can also happen in other cases like the grafting or parasitism. And in parasitism, there are already documented cases for the genetic material or the HGT event happen from the parasite to the host or from the host to the parasite. And we'll talk about the examples later. And in plants in particular, grafting is quite common in some species. And I think you can also see on campus that there's some fig tree and like to climb on the top of some others. And or you can imagine there's some epiphytes growing on top of the another plant. And in this case, there are direct contact of the cells or the tissues from one plant to the other. And in this case, there's some material could be transferred between the grafting tissues. But as long as in most of the cases, even though the genetic material can be transferred, it cannot be transmitted to the next generation since it's not in the germline or in a male stem cell. But in a very rare case, it could be transmitted to the next generation. And there's some evidence for that. And the last, but probably this the most common one is through the vectors. And the vectors could be virus, could be fungi that is connecting to two different hosts, or some aphids sucking up the plants from one plant A to plant B, and or any other insects can transmit it using the virus within the cell. So this is the basic mechanisms, and we already know that this is quite common in many cases, but in general, in eukarya, it's much less common than in prokarya. That's I think it's a general phenomenon. But between prokarya and eukarya, there are many cases has been documented, and so here I categorize into interkingdom DNA transfer, like the bacteria, between bacteria and also the eukaryotes, and also between the eukaryotic cells. And there are very few cases, right, in animals in general compared to the other cases. And we are talking about the plant HGT and plant fungi HGT, and there are cases for fungi HGT, and there's a very nice review paper just published, and so we can refer to this paper.